think we're going to get started. So it's my pleasure to introduce Rasmus Nielsen as our second keynote speaker. Rasmus has long been a star in probabilistic modeling, population genetics, and computational genomics. For me personally, he's been one of my heroes in these areas. During my PhD work, when I first started doing statistical phylogenetics, I encountered Rasmus's name everywhere in the literature, and then I decided to move into comparative genomics, and Rasmus was everywhere in the literature. And then I moved into demography, inference, and coalescent theory, and selective sweeps, and Rasmus was everywhere in the literature. And today, when members of my group and I think we have a good idea, we usually look it up online, and Rasmus has already published it, and usually taken it further than we had imagined. So for decades, he's been an incredibly productive and prolific scientist, but at the same time, something I really appreciate about Rasmus is he's a person who really seems to be driven by curiosity and the desire for scientific insight rather than a thirst for scientific glory. And in that way, he's been an inspiration to me and to many others in the field. Rasmus is professor of computational biology in the Department of Integrative Biology and the Department of Statistics at UC Berkeley, where he's been since 2008, following initial appointments at Cornell and the University of Copenhagen. Through his career, he's been honored with many awards. I won't try to list them all, include election to the National Academy of Sciences, a Sloan Research Fellowship, a Fulbright Fellowship, and election as a foreign member of the Danish Royal Academy of Sciences. I could go on and on about his accomplishments, but I know we'd all rather hear from him directly. So please join me in welcoming Rasmus Nielsen.
All right, thanks so much, Adam. That was much too generous. That's the kind of introduction you get when you're an old fart. And there's some privilege, though, to be an old fart. That is that you can give really boring talks about the history of the field. So I'm going to do that, a little bit of that, sort of from my perspective, and start talking a little bit. There will be some sort of newer results and so on in the end, but I'm going to start a little bit at talking about the history of the field. And when I started in this field, I went to conferences in evolution back in the 90s. And if you went to conferences there, you would see lots of empty DNA trees like this. And you might have 1,000 posters, and 500 of the posters would have an empty DNA tree that they had estimated. And you would then see one tree was in history about how people move from one place to another. Another one was about population growth, and a third one that told maybe a story about continuous gene flow between populations. And the trees were all the same. You could tell sort of any story you wanted from reading the tea leaves of these inferred trees. And of course, what people realized to really get at trying to understand these trees, coalescence theory is what allows you to do that. It allows you to link gene trees, phylogenous estimated within species, to demographic models of selection, and so on. So one of the things I became interested in very early on was how to do that, how to establish that link. And it became clear very early on that really it's a way to do that rigorously and really get the information out of these estimated coalescence trees or mtDNA phylogenes, went through what became known as Felsenstein's equation, which we, with quite a lot of abuse of notation, can write like this. So we have the data x, the parameters we want to estimate y, so this is the likelihood function, and we can think of that as an integral where we weight the probability of, or density of the data given the coalescence trees, or probability measure of genealogies. So we can think of this more practically as a multiple integral over all coalescence times and a sum over all possible topologies of the tree. So that's sort of fairly easy to realize, but of course, how to evaluate this, that became sort of the next big goal in population genetic inferences. And there were basically two approaches for doing that. One approach was to use MCMC, similar to what's done in Bayesian phylogenetics, where you take the genealogy and you perturb it, and you keep doing that, accepting new genealogies according to probabilities that are weighted by this likelihood function, and then run this chain to stationarity, and then also maybe integrate or have the parameters in the macro chain model that you're simulating, and by that getting a posterior for both the genealogies and for the parameters you're interested in. And that became the most commonly used way of addressing this problem and what's being used mostly today. There was another approach, though, that became quite popular for a little while that was based on importance sampling. You basically use what now called SMC approaches to simulate genealogies from some distribution that approximates the posterior. And then using importance sampling, you reweight all the samples that you get, so they're sampled essentially from the correct posterior. So there was a big push on those kind of approaches, particularly by Bob Griffiths and Simon Tavares, that published a bunch of papers of how you might do something like that. Okay, so this is just an example of the result of this. This is a paper much later from about 2015. Beast became the most commonly used program for doing this. Originally, the idea of doing this with microchain Monte Carlo really came from Joe Felsenstein, but this implementation in Beast became what became most popular. This is just one example where the authors of this paper, they looked at changes in population size through time using empty DNA trees and using Y chromosome trees. You get this odd reduction in effective population size for the Y chromosome that actually appears in many different places in the world. There's for humans and many different places in the Y chromosome tree. And it's a bit of an enigma why it's there, but people think it has to do with some massive cultural changes that happened right around that time. You get horses, invent the wheel, and you also get a quite different social structure. And the reason why we think it's not selection is it seems to happen independently in many different places in the tree. That's not really compatible with selection. That's just a result or an example of the kind of things that people today use these kind of methods for. They still exist. And you would use them again for empty DNA, Y chromosome DNA, or maybe for viruses. If you think they don't recombine, you can use those kind of methods. Of course, what happened was that sequencing capacity dramatically increased. In the beginning of the 2000s, you got the human genome sequenced, and you started to get genomic data. First SNP data, and later full sequencing data. And the problem is now you had to deal with the problem of recombination. It was not just one tree, but you have essentially as many trees as there are recombination events in the genome. And as most organisms, including humans, have recombination rates and mutation rates that are at the same order of magnitude, every time you had another mutation, you have another mutation in the genome that can inform you about the tree, you actually get a new tree in average. So these tree-based approaches did not really survive. There are lots of sort of attempts at cutting the genome into shorter bits that might not recombine, but they really do recombine anyway, and then using the old approaches. But many of the approaches that we developed in the 90s and early 2000s, they very fast became outdated. Because, of course, the genealogical structure is now described by an ancestral recombination graph. And I think by now in the meeting, you either don't care about ancestral recombination graphs or you heard about it so many times that you know well what it is. So I'm not going to really introduce ancestral recombination graphs to you, but just remind you that the ancestral recombination graph is a graph where we have two types of nodes. We have nodes that represent recombination events, and we have nodes that represent coalescence events. Embedded in the graph are the gene trees for all the different loci, segments of the genome. And if you know the ancestral recombination graph, you can then deduce the sequence of trees from that. You can't necessarily go backwards because this also has information about where the recombination events happened in the full traditional description of the ancestral recombination graph. So I thought I would talk a little bit about where these ideas come from, because I feel like sometimes a little opaque in the literature. But really all of this is all described in 1983 by Dick Hudson. And I think, to my knowledge, this is the first ancestral recombination graph in literature. And I want you just to all appreciate the mechanical typewriter graphics that he was using back then. Actually sitting, making, I think, slashes with the typewriter to make a graph like this. That's our first ancestral recombination graph, and he described the coalescence process with recombination and the data structure or the object that is the result of that, the variable that's the result of that, the ancestral recombination graph. This later in 1991, where he described a similar model where you have now an infinite sites model, and you have, instead of having individual discrete loci, you have segments of the DNA that recombines, much like we might think about it today. So this is really where the ancestral recombination graphs come from. We often cite this paper, though, from 1997, because it's the first paper that used the word ancestral recombination graph. So that word dates back to there. Griffiths and Marjoram and particularly Bob Griffiths and a bunch of papers of also Marjoram, helped formalize, mathematically formalize ideas about the coalescence with recombination and ancestral recombination graphs, and much of what we know about them today come from their work. All right, so how do we deal with inferences now using ancestral recombination graph? We had the Felsenstein equation before where we had to integrate over all genealogies. What we have to do now is we have to integrate over all ancestral recombination graphs instead. And conceptually it's the same. It's of course computationally much harder. And there was a bunch of different attempts at solving this problem going back in the 90s and early 2000s. Particularly Joe Felsenstein, who led much of the work on how to do MCMC for gene trees. He also works on ancestral recombination graphs and how to do that using similar MCMC approaches. Griffiths and Marjoram, they're also using the type of importance sampling approaches that Simon Tabaret and Bob Griffiths had been promoting. They also developed importance sampling approaches for ancestral recombination graphs. But none of this really took off. And the reason was none of this was really computationally tractable because the object you're integrated over is just so large, this ancestral recombination graph. So I think it's fair to say that people basically gave up on all of this. This becomes sort of largely forgotten and given up on. Instead, when we had genomic data, so we had to give up on this sort of Felsenstein's equation here. Instead, people focused on various summary statistics that you could calculate from the genomic data. The few cases where you could calculate things, the likelihood function, sample size of 2 and so on, or if you reduce the data, have some kind of reduction of the data, then you could also calculate the likelihood function, most famously to use a frequency spectrum, where, for example, Daddy by Ryan Gudenkunst, who is here somewhere, his program is probably the most successful in taking the site frequency spectrum and make inferences based on that, reducing the genomic data to a site frequency spectrum. And often I felt like I was back in the days of taking a single empty DNA tree and sort of trying to make a story over that by seeing many of the approaches that were out there, taking just PCA plots, for example, or mixture plots, and then putting that in your paper, and then you sort of build a big story about what that PCA plot might mean. That these two individuals are far away from each other. That's because 4,000 years ago the ancestors walked from here to there, and so on. You can tell large stories, brew up large stories based on things like PCA and admixture plots and so on. But it's kind of hard from that and then go to rigorous inferences that try to look at how much information do we actually have to conclude that one model is better than the other model, how much support is there for this scenario versus that scenario and so on. And we're back to more storytelling, except for some of these approaches like Daddy and so on, that had the unfortunate consequence of a lot of loss of information, of course, just using the frequency spectrum. So in my head, that was kind of how we were stuck for a long time, and I was very depressed on that until Adam and his group published this paper that showed that, in fact, it is possible to do MCMC or ancestral recombination graphs. And there are a couple of reasons. So basically in this paper, they show you can sample from a well-defined posterior. You can sample ancestral recombination graphs from that. And when first you could do that, that opens up the possibility of doing all of these other things of full likelihood-based inferences, Bayesian and frequentist inferences that take advantage of all the information in the data. And there are a couple of reasons why they could do this. One of them is, of course, a lot of creativity and ingenuity, but also that people realize you can well approximate the coalescence process with recombination using a Markovian process that goes along the genome. And using that approximation, things becomes a bit simpler. And then you had also, for example, the Lee and Stevens copying process that's been proposed that can be used for updates similar to that. And Adam had worked a lot on HMM models for solving various problems in genomics and figuring out that some of that formality, you can propose updates in an MCMC that will actually allow you to make efficient MCMC updates and eventually produce a method that will allow you to sample from a well-defined, well-calibrated posterior distribution of ancestral recombination graphs. There's of course other methods for just estimating the ancestral recombination graph before and after, and some tremendous work has been done on really estimating large ancestral recombination graphs. Some of the contributions are here. For example, T.S. Infer is famous for, or ARC needle, both famous for being able to scale up to UK Biobank scale data, so really, really large data sets to be able to estimate ancestral recombination graphs, and also relate, sorry, this is the Inferring paper, this is the relate paper, relate being able to maybe not scale up to that big samples, but then being able to sample coalescence times, when first you know the ancestral recombination graph, the topology of it, being able to sample coalescence times from a posterior, which goes some way of quantifying uncertainty. I just want to make sure to mention that there are many other methods out there for this, but for me, what was special about ArcWeaver was the ability to actually sample probabilistically from a well-defined posterior. And the reason why I'm interested in that is because when first you can do that, you can really start, in my opinion, to do statistical inferences. And one of the things we want to do is to use importance sampling. A little bit back to the ideas of Griffiths and Tavares in the old days, of sampling arcs from some well-defined distribution and then rewriting those arcs so that with importance sampling we can get a distribution of arcs, of weighted arcs, that then represent a sample from the distribution that we're interested in. And if we can do that, then we can make inferences under many different models. And one of the first things we started to work on is how to do that on selection. And I'm going to talk now a little bit about some of the work that we did on using ancestral recombination graphs for inferring selection. And this work by my former student, Aaron Stern. And the idea is that you want to use the path of allele frequency change through time. We know that if you know that path of allele frequency change, that is sufficient for the selection coefficient, that is all of the information about the selection coefficient, about selection, is in that path of allele frequency change. We also know that if we can, using all of the data, infer the gene tree in a selected locus, assuming that there's just one selected locus, then that contains almost all the information in the data about selection. There are back recombination events that can add a bit more information that we're ignoring, but almost all the information is in the gene tree. So based on that kind of logic, he devised a method where the two hidden layers that we're integrating over, two sets of latent variables. One is the arc, that's different trees in the different positions in the genome, and the second layer is the allele frequency path through time. And this one, we could deal with the hidden layer one by using ArcWeaver to sample ancestral recombination graphs, and then we could use something similar to standard hidden marker models to then numerically integrate over the allele frequency path. And putting that together, we can then do maximum likelihood inference of selection coefficients, for example. So this is the kind of things that you get out of it. This is some data simulated where there's no selection. This is where there's a bit of selection. You get a likelihood surface, and you can infer the selection coefficients pretty well. And do then likelihood ratio test, various forms of hypothesis test, model choice, and so on. And what you also then get with it for free is the allele frequency trajectory back in time. So this is, again, the example with selection and without selection. With selection, there's been a strong change in allele frequency. Without selection, there hasn't. And the coloration is a posterior for what the allele frequency was back in time. Okay, so that's implemented in a program called CLUES. And this is just an example of that. This is the lactase locus, which several people have been mentioning previously, one of our most celebrated examples of natural selection in the human genome. This is in Europeans, where selection has favored lactase persistence, so you can drink milk as adults. And what you have here in the black line is what's inferred by CLUES of allele frequency change. And then we have here in red from Madison and Madison, the allele frequency also for European populations inferred from ancient DNA back in time. And it sort of matches pretty well, although you can see maybe it's a little off here for some reason. And for a long time we thought maybe that's just a matter of our modern samples representative of the same ancestral samples as they're analyzing. But I think maybe, and I'll get back to that, maybe also something else is going on here. You can get confidence intervals then, parameter estimates with confidence intervals, and this is a new version of the program we have made by Andrew Vaughan here, who implemented a much faster and solved some of the numerical problem in the first version of the program, CLUES 2, that represents that, and this shows estimates of the selection coefficient using clues 2, and then if you try to make actual likelihood estimation based on various summary statistics, we always like to compare to Tadimas D, right? Because that makes everything we're doing look so good. But this is sort of the distribution of the estimates around the true value of clues 2, and just shows that, not surprisingly, if you have a full likelihood method, you can do better than if you just use a summary statistic. That should not come as a surprise. What Andrew also implemented was a method so that you could allow different selection at different times. And one of the great things of working at the ARC is that you have everything time labeled, so now you can really get at issue about how things change in time. And so you can do model choice for some of the loci that shows most evidence of selection. Is the evidence of changes in selection through time, or is selection more or less, has that been constant through time? And this is an example of that, and I'm going to point out here the MC-M6 locus that sits up here, and here, and here. This is a model with 1 epoch, and with 2 epochs, and with 3 epochs, and then you can calculate AIC, and we can see that we get the best AIC score in a 3 epoch model. MC-M6 is where the causal variant for the differences in lactase expression sits. So we know that the causal variants sit here, and that's what's presumed causal variant that we look at selection at. So what we're getting at here is that there's statistical evidence that, in fact, selection on lactase was not constant in time. And that's something that other people have been arguing before us, that it doesn't seem like a model of just sort of the same selection acting everywhere fits really well to the lactate locus. And this is the path that we get when we allow different selection coefficients through time. And what you see now, we have a much lower frequency back in time. And what the data seems to suggest is that, at some point back in time, there wasn't really any selection on this locus. And in fact, I think this should hit 0 here, this path, and I think that what it doesn't go, we actually have some old genomes that infer to have the causal variant, and I think that's an error because there's doing genotype calling that is imputation based, and you have the old haplotypes that haven't received the mutation yet. And so then you sort of see them, ghost mutation in the past that's not there when you use that, because today everybody that has that haplotype, they have the mutation. So with high confidence, the imputation program will say, okay, the mutation was in that haplotype, and override, if you have 2 free reads in the ancient DNA data, it will override that information saying, we believe that the mutation's there. So that's why I think it doesn't actually go down to 0. But it seems like there was really strong selection in the period from about 5,000 years ago to about 2,500 years ago, and then less selection here. And it corresponds, there was a wonderful talk previous in the meeting, now unfortunately I forgot the name of the speaker that gave, but wonderful talk about selection on the lactase locus that also mentioned this correlation with pastoralism. And this is a period where in Northern Europe, pastoralism had sort of replaced an agriculturalism. Instead of growing cereals and relying mostly on that, they were using dairy farming at this point in time to a much higher degree to get food. And that changed again then about 2,500 years ago. So it tracks that quite well, that the strong selection is associated with dairy farming and a life of more pastoralism. Okay, so this is selection on a single locus, and we like models like that because they're easy to deal with. You heard a lot about polygenic selection already at this meeting, and we're not going to ignore that too. Really, of course, selection is acting on a phenotype, and with this very rare cases, as perhaps maybe lactase persistence, but it's very rare cases where selection just is acting on a single locus. Typically, multiple loci will be associated with selection on a phenotype. So we can also, on a very simplifying assumption, incorporate that into the model. And Aaron Stern implemented a model looking at this that he called PORM, that basically is assuming that you have directional selection and then what's called the Landier approximation, a very simple approximation that basically you have additive, you add up all the effects. You can then look at, based on the evidence of selection on each individual locus, the joint effect on the trait. So now you can estimate a selection gradient, the selection acting on the trait, not the selection acting on the individual SNPs. And if you do that, then you can then test if there's selection on a trait, and we can show you have quite a lot of power. This is a method that can deal with hundreds of thousands of samples. We can only do hundreds of samples, so maybe the comparison is a bit unfair here, but it just, it's a nice method to compare to if you want to show you have a lot of power. That's what we did. There is another existing polygenic selection method that doesn't compare populations, but just looks within a population. So we applied that to some real data, UK Biobank, effect size estimates, a collection on that, and here is just looking out in the tail. What are the traits where we found most evidence of selection? One is called tanning, so that's really a proxy for skin pigmentation. And another one is, for example, here is a phenotype that's associated with type 2 diabetes. And these would be the ones where we find strongest evidence of selection. Okay, so all of this has ignored another thing, that is pleiotropy. So, of course, like multiple loci will affect the same phenotype, some loci will also affect multiple phenotypes. It's probably very highly likely that most traits are quite pleiotropic that we could be looking at. So that creates genetic correlations. And one of the interesting things is that we can also look at how selection affects then that action one trait might affect the other trait. And here's an example of that. So there's long in the literature been a claim that there's selection for hair and eye color in Europeans and then sexual selection. So the idea here is that people that are blonde are more sexually attractive, and that has favored blondness in the population. A lot of anthropologists doubt that, and the reason why they doubt that is that they would argue that what tends to be attractive is what the ruling class, the traits that they have. And that's modeled, you model attractiveness after the rulers. And perhaps in some part of the last 200 years, there have been periods where many places in the world that blondness has maybe been attractive, but that's just really a consequence of the fact of European colonialism, that they were the rulers, and therefore the traits they had sort of became what was considered attractive. But some papers argue that we are like peacocks, that selection favor, certain hair colors and so on, that there's sexual selection also acting in humans. So interestingly, if we look at hair color marginally, we also get evidence of selection acting on hair color. But if we condition to a joint test and we condition on the genetic correlations with skin pigmentation, the evidence largely go away. So what we think is going on here is that what people thought was selection favoring hair color was just correlated selection associated with skin pigmentation. And the skin pigmentation, we understand very well, of course, what that is. That is, if you are in a climate where there's not very much UV radiation and you have a lot of skin pigmentation, you're more likely to suffer from vitamin D deficiency. If you're in a climate with a lot of UV variation and you don't have much skin pigmentation, then you're much more likely to get skin damage and skin cancer and so on, other negative effects. So I think the skin pigmentation story we understand quite well, we think at least, and I think the hair one might have to do really with correlated selection, that people that became blonde in Europe, that was just correlated selection with other alleles that favored a lighter skin color. It seems like, at least from this. Okay, so a big question with this is how well do we actually estimate arcs? Because all this rely on that what Adam and his group has been doing, that actually really works for us. And if that doesn't really work, then we're not doing so well. So we started to do a lot of simulation studies of looking at various properties of arcs. And this is an example here where we compare ArcWeaver and Relate and TS-infer plus TS-date. And what you have here are the simulated coalescence times and estimated coalescence times. And we look at the correlation here between the true and the simulated value, the true and the estimated values, and also the mean squared error. And you can see here that by this measure, ArcWeaver performs reasonably well, much better than Relate, that performs substantially better than TS-infer and TS-date. Okay, but what we then thinking about was if perhaps we could do something that scales a little bit better up than ArcWeaver, because while ArcWeaver solves the inference problem fundamentally, it doesn't scale up to hundreds of samples really, and we would like to go there. And that's something that Yun Deng worked on for years and years and years. He was sort of thinking about scalability versus accuracy. ArcViewer lying here, things like TS-infer with TS-state and relate lying there. And he wanted to see, can I do something that's more up here, higher with accuracy that can compete with ArcViewer, but be able to scale up to larger samples. And he had a bunch of different ideas for how you could do this. And one of the core ideas is to separate when you do the MCMC, to separate the steps that finds topology from the one that finds branch length. So when you take a new edge and you thread it into, you're trying to figure out, you pull it out, and then you try to figure out how does it fit back in the arc. He developed an algorithm where he first determines where in the arc, when the topology of arc should it fit, and and afterwards he then sampled the branch length. And that turns out that's an approximation. It's not exact, but it turns out that there's not much of a cost of that approximation, and it can really speed things up because the hidden Markov models that you look at can work well with many, many fewer states, so you reduce the state space. And he also did other things. He did, and there's an update of internal nodes that's really based on, or inspired by one of the updates going back all the way to Felsenstein's work that Felsenstein did with his postdoc Mary Kuhner. They had a specific update that he somewhat mimics for moving internal edges in the arc around. And the result is something that works that's pretty fast. These are some comparisons here in seconds. I'll show some more soon in terms of arc size versus time, computational time needed. And accuracy still looks good, so this is mean square error in allele age is estimated for 50 sequences and 300 sequences, comparing Singer, as he called his program, to relate and TS-infer plus TS-state. And this starts looking a little bit about how the MCMC mechanics. This is then ArcViewer, and this is Singer, and you can speed up so it converges much faster using some of the tricks that he used, and also make the updates faster. So all of that we sort of conclude that there's a method now that allows at least to scale up a bit further from what ArcViewer could do without a loss in accuracy. So here's some other benchmark on mean square error and pairwise TMRCA estimation. We added ArcNeedle in here to the bunch. We have relate and TS-infer and ArcViewer here for 50 sequences, and we think at least in these cases we seem to do better than ArcViewer. And I think there could be many reasons why that could happen. Could be the discretization, but I think it simply has to do with convergence issues that it's just hard to get ArcViewer to really converge fast in these repeated simulations. So it seemed like it performed quite well. And if you look at topology, so this is the branch length. This is in terms of topology. How many errors do you make in topologies when you count triplets? And again, Singer seemed to perform well. But more importantly, it still samples quite accurately from the correct posterior distribution. And one way to look at that is to use a posterior predictive check where you simulate a bunch of data sets, where you first simulate parameters from the prior, and then you simulate arcs from those parameters. And then you look at various statistics, for example, node times to most recent common ancestor, and so on, and compare the estimate, the true one to the posterior distribution. And then you can prove that statistic, that rank statistics, should be uniformly distributed if the method is calibrated correctly. So if it's a valid Bayesian method. And I think it's an important check. We often, when we do MCMC, we check, does it converge? And we check, okay, do I get the prior distribution back if I run it without data? But this also checks that, in fact, the likelihood function and the interaction with the likelihood function works. And what we can see here is that Singer very well produces this kind of well-calibrated posterior distribution. And if you look at credible intervals, they also look pretty good for Singer. 90% credible interval here, we get at 85% coverage there. 50% credible interval, 47%. So it seems to be well calibrated and provide a valid posterior distribution of ancestral recombination graphs. So here's just what you could use it for. This is again for the MCM6 gene, that's lactase again. You can look at the degree of variability along the sequence if you look at all samples in Europe, and if you just take the ones that carry the derived allele. And what we can see here is what people have seen with other approaches as well, is this complete depletion of genetic variability. Basically, the branch length, if you look at the branch lengths, they're essentially shortened to be almost zero for that, because there's been this strong partial selective sweep in Europe that has wiped out all the variability, and then you have basically these zero-length branch lengths. You can also look at balancing selection. So this is HLA region, and you can go along the HLA region and look at the time to the most recent common ancestor in different parts of the HLA region, and you get some of these coalescence times up to 25 million years, compatible. We know there's strong balancing selection. But I should maybe say one other thing. One of the advantages of using ARCs for this kind of stuff is that traditional approaches will take a sliding window. And you will basically take a sliding window that sort of averages out over many of these things. But you know, for balancing selection, the signal should be incredibly short, maybe just 1,000, 2,000 base pairs if it's really strong balancing selection. That's because there's tons of recombination events that can happen in those, say, 25 million years, where selection has been acting on the variant. And using ARCs will allow you to get this kind of detailed picture of it, and will associate a measure of credible intervals. If your word is at 1 peak or 2 peaks really, well, you have measure of a statistical uncertainty that can tell you that. Here's another gene. You can look at expected time to the most recent common ancestor in all the data versus how much that's within, to look at where is there depletion of genetic variability within a locus, maybe due to a selective sweep, and here's just an example of that, the kind of signal you can find with that. Now, here in the end, I'm just going to talk about one extension that we've been working on that recently, also Unden's work, and that's a program called Polygon. And the idea in this program is to develop a method for better calibration of the coalescence times in the ARC. And in particular, to try to use an improper prior for the coalescence times. Now, of course, all of you will know from coalescence theory that you have an exponential distribution of coalescence times. The reason to use an improper prior, there are several different reasons for it. One of them is then the ARC really gets driven just by the data, not by the prior. You might like that if you're not sort of super Bayesian, or if you don't really believe much in that exponential prior. But there's another reason for it. These kind of important sampling approaches, where you take the gene analysis and reweight them, they will tend to perform much better if you use an uninformative prior than an informative prior. So for that purpose, this is really useful. So he implemented that. But he also did another thing, because he was realizing that the uniform prior, if you truly have a coalescence prior, doesn't perform as well as using the coalescence prior. That should not come as a surprise. If you use the correct prior, you'll get better results than if you don't put that information in. But his idea was then that, well, we can maybe rescale our results so we get the same results as if we had used the correct prior. So basically what you could do is a posterior predictive check, where you look at each tiny interval, how many mutations are occurring in each tiny interval. And if there are too many mutations in the time interval, you know that you should expand the time there. If there's too few, you should contract it. This seems maybe like an odd thing. We say we have a sort of misspecified posterior. They misspecified because the prior was wrong. And then we can make a correct posterior. And if you're Bayesian, you might, like, there's no such thing as, an incorrect prior. The prior is, my prior belief about how it should be. But one way to think about what's going on here is very similar to various empirical Bayes methods, where we have a problem here where every time we add another edge in the tree or another site, we sample again from the same prior. So we don't have things, the whole distribution will not converge to the true distribution. Okay, so it's not like usually Bayesian inference you would want the likelihood to overwhelm the prior, but it doesn't happen here because we make a new sample and a new sample and a new sample where we add information from the prior and from the likelihood at the same time in a way so it doesn't converge. But you could use this rescaling step to actually correct, so the posterior you get is the posterior you would have gotten had you sampled from the correct prior. But you don't have to sample from the correct prior to start. And we can show that that actually works using these uniform distributions. This is on a constant model of constant population size. This is a CU demography, this Rubin estimated demography. And the red line is what you would get if you just take what you would get if you don't do this rescaling, and the blue is if you do the rescaling. And what you can see is, for this order statistics, that if this is a well-calibrated posterior, it should be uniform. It's very, very close to being uniform. So we actually get out the posterior for the arc that we should have gotten had we known what the correct prior was. That's, I think, a little bit remarkable. You can sort of bootstrap yourself up, figuring out what the prior should have been, and then get something that's still a valid posterior. Okay, so this is a little bit about how that performed. We actually get, particularly for these other demographies, but also even for constant population size, we can improve the performance on Singer quite a lot by using this cool divergence from between the true distribution and the estimated. We can get quite an increase in the accuracy of the estimates, for example, of pairwise GMRCAs. And also use it, for example, for estimating effective population size. This is for a very simple method that just take the pairwise coalescence times and no penalization or anything else. And you can see here in blue you have Singer, and in black is the ground truth, and they track pretty darn well and arguably perhaps better than some of the state-of-the-art methods that are out there for estimating NE. And that's without thinking about penalization and anything else like that. All right. It also has another effect, that is, for example, if we go back to the MHC, I said you get coalescence times that are 25 million years old. Actually, now you get more like 35. And why is that? Because you don't have a strong prior that pulls the estimates towards more recent estimates. So our best estimates is that actually in some of the places in the HLA region, the coalescence times as old as 35 million years. All right, and so I will end with that. I want to make sure I thank everybody that have been involved in this. Particularly, I didn't mention Yun Song, who co-advised Yun Deng, and also Will DeWitt, who has been involved in this project, and other collaborators on the selection part of it, Noah Seidlin, particularly, that helped us a lot thinking about genetic correlations and how to deal with correlations. And thank you again for inviting me and for having me here and for listening to me today.
Really cool, interesting. Just a question about how selection at a site could impact the inference of trees, of ARGs or trees at that site. Assuming a neutral prior will probably push everything back, but selection will probably push it closer to the present, and whether this polygon could help with that descaling.
Yeah, I mean, in theory, so that's correct, right? So that because you have a prior that assumes neutrality, if you have a strong sweep where branch lengths are really short, it will sort of bias your estimates in that region towards longer branch lengths. And I do think polygon can help with that, but the important sampling method, it directly deals with that, right? Because it knows that, so it calculates, what should it have been if it was actually sampled under a selection model. So these important sampling weights, so calculate what's the probability of sampling under one, or the probability of the genealogy under one model and versus the other model. So it addresses the difference in prior. In some sense, that's the information it uses, that there should have been another prior in this region, and that's the kind of prior you would have on a selection. So particularly for these selection inferences using clues and clues 2, then that is incorporated in. Oh, I forgot to say another thing, sorry. To answer a question that hasn't been asked, couldn't you do this couldn't you also improve in the same way you do clues 2 with selection multiple time intervals, couldn't you also do that for polygenic traits? You might ask and say, yes, good, thank you for asking that question. And Andrew Vaughan has a poster about that I just wanted to advertise for in the poster section that come up. So please find Andrew's poster about that. All right. Sorry about taking your question and moving into something else. I just realized I'd forgotten to say that.
Hey, I'm wondering about your thoughts on sort of the difference between using the really big data sets where we can't get, well-calibrated posteriors, but maybe if we had the posteriors, they'd be narrower, and smaller data sets where we could use Singer and, use an actual model and get an actual posterior.
Yeah, so do something combined, like take some information from something like Singer, but then also taking TS infer to sort of see, can you scale up and can you relate the two to each other, and so you can use the larger samples. Is that the idea?
That would be great, but no, I was asking a simpler question, which is like, both these things seem useful, and I'm wondering about your thoughts.
Oh yeah, both are certainly useful. I mean, all the the lots of talks about analyzing UK Biobank data and so on, and Singer will never analyze UK Biobank data. Oh no, so so I think certainly there's space for both very much. And what Singer, maybe I should say about what it can scale up to, I think Singer can do a few hundred sequences, and maybe if you're patient, maybe up to like 500 sequences, but I don't think in the current form, it will be able to do much more than that. And of course, much of what you're interested in human genetics is 500,000 sequences, and it will never be useful for that. Yeah, so if the question is, is there room for both? Certainly. There are lots of things that Singer cannot do.
Hi, Rasmus. Nice talk. I had a sort of a philosophical question. It seems like basically we've gone from trying to solve a hard integral to basically placing a point mass on one thing and then doing inference with that, which I've always been a little uneasy about as a statistician. And I wonder if that's ever bothered you, if you've thought about it in that way.
In which way do we put the point mass on one thing?
Well, I'm thinking more about, I guess, TS infer style of inference. You sort of get one big tree sequence, and then we do lots of downstream inference, but we've seen that that has many problems for inference in general. Like there's lots of ways in which that can go wrong, and you have poorly understood biases that creep up, and it's really unclear how we correct that. I guess the Bayesian method is a bit more robust to it, but sort of maybe can't scale to this data that we actually have. So there's sort of a tension, sort of like what Peter was saying. I'm just curious if you...
Yeah, I mean, maybe I don't fully understand the question, but let me give you an answer anyway. Perhaps it's an answer out of ignorance. One thing that I think we can do in this framework is to look at posterior predictive tests, right? So we can look at how well does it actually fit the data. In some sense, that's what we take advantage of with this recalibration. And I think that there's an advantage at being able to have full probabilistic methods where you can actually say, does this really, fit the data, and are our credible intervals, do they have the right, the right size, and so on. So I think that's, certainly, I mean, do we make assumptions that are wrong? Certainly. I mean, if you're in this room and you're population geneticist, you're welcome to the club, right? I mean, we all do that. But we make strong assumptions about exchangeability and so on, but I don't think maybe they're worse than what I've been used to do for the past 30 years. I don't know, and what other people are doing. I don't know if this answers it. But maybe we can talk about it later.
With all of these methods, we're still sort of restricted to regions of the genome where we have one-to-one orthology and collinearity and all of that. Do you see any foreseeable future where we start to handle copy number variation and duplications and rearrangements and that sort of thing with ARCs, or is that something we shouldn't even bother to try?
I mean, I hope somebody will do that and solve that problem. But have a pan graph that's also an ARC and so on, and can deal with the structural variation. We thought a lot about it because we're interested in analyzing some inversions, but even really analyzing inversions, you sort of loop around yourself, right? In a way that we can't really accommodate in the ancestral recombination graph. So I think that that's a really important question, and I don't have an answer, I think. Maybe you'll solve it for us.
Yeah. So just commenting on that previous question, is there particular areas of the genome that ARC inference is really difficult, aside from these?
Yeah, I mean, I think anything where there's structural variation, right, that could cause problem in some sense for any population. When you can't figure out what's homologous and what paralogous, I think everything that we're doing usually that will cause problems. And I think the major, right now, I mean, I should mention also other caveats. This relies on phased genomes, for example. And the kind of sample size it's applicable to is really very much for non-model organisms, not humans. Humans, we have hundreds of thousands of individuals, right? But we really want to, look at all kind of non-model organisms with this, but it requires phased genomes. So one of the things that UNIS is working on these days is trying to figure out, okay, how bad is it, phasing error as a, how does that affect the ancestral recombination graph? And so that's a big problem. And of course, the structural variation, it just can't deal with that, I would say.
All right, I think we'll stop there. Let's thank Rasmus one more time.